এই তো জয়েন করেছি তো।
হ্যাঁ দাদা তো
তুমি থাকবে নাকি জয়েন করার পর্যন্ত?
আমি স্যার পুরোটাই আছি, আমি বাড়ি আছি স্যার, কোনো সমস্যা নেই।
ঠিক আছে,
আমি আসি পরে আছি আসলে। আমি তো বাড়ি থেকে দেখবো দায়িত্ব। ঠিক আছে, 11টা 5 মিনিট আছে মাত্র। ঠিক আছে, রাখলাম। হ্যাঁ আমি বাড়ি আছি, আমি 11টার সময় করে আসবো।
I
think we are live, and if Professor Basu, you're okay, we can start.
Yes, I'm okay.
Yeah, okay, great. So good morning, everyone, and welcome to this first webinar of the iBreak Plus Biotech Spotlight series. This webinar series is being organized by all the iBreak Plus institutes to inform the public about the key scientific outcomes from our research. So, as the first talk from NIBMG, we are very pleased to start this series with research from Professor Anil Basu's lab. Professor Basu's research interest is in understanding population genetics, genetic epidemiology, and admixture mapping. Today, he's going to talk about a very recently published work from his lab, which was published in Nucleic Acids Research Journal, titled Deep Learning Insights into the Distinct Patterns of Polygenic Adaptation Across Human Populations. So without taking too much time, I would request Professor Basu to please take over. You are muted.
Good morning, everybody, and thank you, Alpana, for your introduction. I guess I'm audible now. Am I audible?
Yes, you are.
Okay. All right. So I will start sharing my slides. Let me share my slide. Can you see my slides? Can all of you see my slides?
Yes, they are visible.
All right. Okay. So I'm going to talk about this. It's a great pleasure to talk about this work that we have recently done in this Spotlight series, and my name is Anil Batu, and as Alpana said, I'm a professor at the National Institute of Biomedical Genomics. Below this slide is my email address. So just in case if you have any specific questions which you haven't been able to answer or a query about this paper, you can write to me. So this work has primarily been the work of Devashish Tripathi, who is a fourth-year PhD student. This is a work for his thesis. He is doing his PhD in biostatistics in the GSK, RCB, and IBMG joint program. Besides Devashish, this work is also also tireless efforts of Dr. Chandrika Bhattacharya, and this has recently been published, as Alpana has mentioned, in the nucleic acid research. So before I get into this, I would like to tell you two things. One is that raising, what we have done for raising is we have developed this algorithm and we have applied this model on human genetic data, but this can as well be applied on all kinds of data sets for any organisms, and it has much more flexibility than what it has been applied on right now. So the basic model development, so there are two components of this. One, of course, is the model development, and there has been substantial improvement in the model that we have developed over the existing models. And some of these concepts, some of these model development concepts can easily be applied to other kinds of problems where people would like to use artificial neural networks to solve their own kind of problems. Raising is also available in a GitHub repository and also in Zenodo. This is publicly available. If you have a problem in mind, or if you like to try our program, please download it from these websites and use it. So we'd like to hear what your experience is about using this program. As a prelude, we'll start with, as I've said, I mean, my interest is in human genetics, and we work in a lot of human genetics. And as a prelude, let's talk about the peopling of the globe, because this will be primal when we apply raising in real data. And eventually, the test of any model that you develop is how it performs on real data. So if you think about our understanding of modern human evolution and migration, so anatomically modern humans, they evolved somewhere in sub-Saharan Africa, and that happened at least about 200,000 years ago. It's only about 50 to 65,000 years ago then where the first successful movement out of Africa actually happened. So the hypothetical migration route of this early migrations are in dark green, and the first migration possibly was in this Levant region, and from there has been movements towards Europe, movements towards East Asia, and as well as very early movements towards Southeast Asia. Subsequent to that, as the green lines becomes yellower, are the more recent movements and the more recent places where people have moved. So the gray places are where there has been evidences of expansion, and there has been which are primal. So there are two things which need to be kept in mind here. One is that we lived in Africa for a very long period of time. So for at least more than half of the time we are on this earth, we have been living in Africa. And then there has this first movement has been of a very small number of individuals, which we know now from various other studies. And this movement has been very rapid in terms of people moving. And this has really been an exodus where people move to different parts of Eurasia, as well as to Australia in a very short period of time. All right. So, given that background, let me look into something else, which is the skin pigmentation map of the world. So, if you look into the skin pigmentation map, this is something which is like a depiction of the average skin color, something like an average skin color. So, higher numbers represent a darker skin color, and these are the different continents. And as we all intuitively know, that people who live near the equator, people who live in the torrid zone, so to say, within the Tropic of Cancer and Capricorn, they generally have darker skin color. And a reason for this has generally been assigned to adaptation of human skin color, and this adaptation has given us some kind of selective advantage, be it in vitamin D suppression, vitamin D absorption, be it in protection to different kinds of possibilities of ultraviolet exposure, which can lead to cancers, etc., etc. So this is intuitive. This is well known that this has been an adaptation with respect to something. Now, how does this adaptation, which has happened, pass on from one generation to generation? Obviously, this has to be somewhere hardwired to our genetic information. So our genes of the genes of populations of individuals who live around the equator typically have alleles which have high melanin secretion, which gives rise to dark skin color, and that is passed on from generation to generation, which gives these populations their signature skin color. So this is true for an example of skin color, which we intuitively know, but this is possibly true for everything, many other things. So this is the same graph where you on your right you'll see the average temperatures in January, the average temperatures in July. As you can see, places where it is hotter typically have darker skins. So what we are doing in a sense is we are responding to changes in the environment, or we are responding to our environment, and that adaptation is somewhere hardwired in our genes. Now, the skin color is related to the average temperature of the region. We can translate to that skin pigmentation is an adaptation in response to temperature. Or if we try to formulate, then we can also think of skin pigmentation as a function, in some ways, a function of temperature. So it is almost like that there is a relationship between the temperature and skin pigmentation, which is some kind of a functional relationship. Now, this is not limited to temperature. Of course, we have been living in different kinds of habitats, and there has been different kinds of adaptations which we possibly have to undertake in living with different kinds of habitats. And different kinds of habitats give rise to different kinds of challenges. Somewhere it is hypoxia, where the oxygen is limited. Somewhere it is living into a desert where the environment is very different. The mangrove forests are living in seasides. But there are other kinds of issues which are also related to this, which is like there are pathogens. The pathogens are different in different kinds of environments. So your adaptation is not necessarily to the external environment, but also to the pathogen load of the specific environment. All right. So we optimize to our environments. Now, the challenges that has been posed to us are not necessarily geographic, which varies with environments, which varies with altitude, etc. There are other kinds of challenges which are like temporal. So, for instance, if you look into this picture, this is present time, and as you go towards your right, you move towards past, and this is temperature deviation with respect to what we see is now. So this is our temperature at present. And if you think of this, then only about 22 between 20 to 10,000 years ago, our Earth has started warming up. Before that, the temperature was cooler. So there are certain kinds of changes like temperature, which we have already convinced you in a sense that that has a huge effect on our adaptation. There are changes which are temporal in nature, the changes which changes with time. So we do have to adapt with changes which has happened long back, and and in order to move forward in evolution, we have to adapt subsequently to different kind of temporal changes as well. So there are different kinds of selection pressures or selection gradient which are applied on us. Some of these are spatial, like geography, altitude, pathogen exposure, which is related to that. Something which is temporal. For instance, there can be pathogens which are temporal, comes and goes. There can be pandemics, which can be results of this. Some of these can also be anthropogenic in a sense that suppose if you when you start agriculture, you have exposure to different kinds of pathogens where you didn't have exposure. When you start agriculture, the population size expands, so there are infectious agents which are now being transmitted easily. If you start agriculture, there are changes in your diet, changes in your nutrition profile. So there are changes which are temporal as well, with which you have to cope into this. So the spatial changes and the temporal changes are in some ways interlinked as well, because some of these spatial changes leave specific kinds of temporal changes. Because suppose for example agriculture, the agriculture depends on the geographical region. What kind of crops you cultivate depends on the geographical region, and it is something which changes over time. So there are relationships between spatial and temporal, and they have overall a joint impact on our selection or on our genomes. So how do we look at the genetics of adaptation? So there are models for this. One, of course, is what is known as a monogenic model, which is that suppose if you look, it is also sometimes typically related as roughly as something which is like a hard sweep. So if you imagine these blue lines are suppose our genomes, and these genomes before selection, these genomes are all blue. And now suppose there is a mutation which gives us some kind of a selective advantage, which would mean that this mutation will rise in frequency in this population, because if you have this mutation, you have selective advantage. So you reproduce with more efficacy, you reproduce with more success. So there will be more and more individuals in the next generation with that mutation. And as it goes on, eventually that mutation almost fixes itself to the population, which means almost in some cases it's like 100% of the population has this mutation. And not only that mutated position which have this causal effect, but the region around that has some kind of a hitchhiking effect, which is like they are linked to one another, which is like a long haplotype which typically gets selected. So there are examples of these kinds, like the Duffy allele, which gives protection to cerebral malaria to Africans, has something like a close to 100% frequency, 100% allele frequency in Africans, but it has very, very different kinds of frequencies in non-Africans overall. So this is the hard sweep model, but her life is not that easy over times, because most of the traits which gives us the effectiveness to find these kinds of selective pressures are polygenic in nature, which means that the mutations are not necessarily on the same region of the genome, but they arise in maybe independent regions of the genome, which may be very, very distant. And sometimes these mutations are present there and they do not raise to 100% frequency. And sometimes new mutations also sometimes rapidly give rise to changes which or advantages which were not there, and none of these gives rise to none of these gets to 100% frequency, but typically all of them increases to a certain frequency. So they increase in frequency but never gets to a fixation. And this is something which is related as a soft sweep, as we understand that most of these complex traits by which we provide ourselves with this adaptive advantage are necessarily polygenic. So which means that it is not so easy to find out this because they are related to one another. They do not get into 100% frequency. The whole challenge is to find mutated positions like these mutations from this entire huge genome, which is much more difficult than finding a few needles from a huge haystack. And you never know how many needles are there. You do not know how long is this needle. You do not know about anything about this needle. So this is a relatively complicated scenario, a much more complicated scenario than in the previous case, where you know that there is one needle and you know the length of the needle, and the length of the needle is pretty big. Whereas this is not like that. There are many needles, and the needles are related to one another. So what we try to do is that we try to have different kinds of mathematical models by which we try and find which are these genes with respect, or which are these mutations which gives us this kinds of selective advantage. Now, so this gives rise to our research hypothesis, which is that the genotype and the environment, they interact, and this gives rise to specific patterns of allele frequencies. And the whole idea is to identify these patterns and in our genomes. So our objective was to develop a deep learning architecture for detecting signals of natural selection. Now, I get to this. I'd like to, before I formulate this, I'm not going into the details of the mathematical formulation, but I'll just give you an intuitive understanding of how the mathematical formulation takes place. So the allele frequency of the dark skin will decrease as the distance from equator increases. So let's come up with a hypothesis of this kind. I mean, if we think the temperature is impacting some kind of a selective pressure, which is going to change your allele frequencies for the dark skin allele, then the allele frequency of that dark skin or the high melanin content allele will decrease as the distance from equator increases. So if we think about the allele frequency as y in this graph and the distance from the equator of a population as x, then there can be some kind of a relationship. Here we have taken a simple quadratic kind of relationship, but it is an unknown relationship that it will typically have. And it's not going to be as simple as this. And there can be bumpy relationships here. But any allele, which is any allele or any gene or a locus where you have an allele which has something to do with melanin secretion or your dark skin, will have some kind of an inverse relationship when the distance from the equator increases. So your allele frequency y will be some complex function of x, where here you can think of x as the distance from the equator. Now, any allele which is not related to skin color, that those alleles will not vary like this, and they will vary in a random fashion. So other alleles will vary in random fashion, whereas the allele which confers to a which confers to a dark skin will have a specific kind of a relationship. So if you study multiple populations, there is a possibility, a theoretical possibility, that you can find these strains of these clients in these alleles, and in order to in finding these clients, you can try and relate them to those specific kinds of changes with which you would like to relate them. Now, it is obviously a important question that the selection gradients are not necessarily linear. So just to sum up things, so if suppose the selection gradient, which is on the top panel of this figure, varies like in a strange fashion, that there is a selection gradient. So this x-axis is like your geographical spread. Just imagine that everything is spread in one dimension, and the selection gradient on this particular region is high, and the selection pressure on this particular region is low. So if you look at different populations living in this spread, then populations which live along here, the allele, the advantageous allele, which is depicted in this orange color, will have high frequency, whereas the other alleles will randomly vary. So if the selection pattern decreases in this fashion, then this allele will also kind of have a decrease like this. If the selection pressure is strange like this, then it becomes the most difficult place where you do not know how to delineate this orange from the blue. And this is where the challenge really comes, where the selection pressure is non-uniform, and the selection pressure is, and this is not only for one allele, but there are multiple alleles, and they're somehow related to one another. And we use all of this information to our advantage to try and find those advantageous alleles. Okay, so what is common here in artificial intelligence, machine learning, or deep learning? As you see that we have employed a deep learning architecture in trying to find this out. So what is important here, or what is common here, is essentially what is also common in regression. So what we have is as we have said, that the allele frequency y have some kind of a functional relationship with x, where so your x, so the independent variable, can be multivariate. So there can be various kinds of things like geography and whatever constitutes environment. It is also sometimes temporal. So x can also depend on t. And some of these may come, some of these may go, some of these may vary over time, etc., etc. And it is obviously some of these are unknown. And then you have your y. In this case, y is some kind of an allele frequency, which is an output or a dependent variable, because it is eventually impacting on your genome. And this x and y are related, but we do not know the exact form of the way in which this is. So what we try to do is the general formulation of artificial intelligence, machine learning, deep learning, or regression is trying to find how this chi behaves, so that we can, if you have an x, then you can also predict what y is. Or if you find some kind of an y, then you can find which x gives rise to this kind of an y. So we want this chi. Now there has to be a note of caution here. One is that this relationship, which we thought is like when we, I mean, so one way, I mean, as we are thinking of this, like the temperature equator kind of a relationship, we could use something which is like a linear relationship. But as you see, the allele frequencies are not really random in populations. So if you have populations whom you collect from here and a population whom you collect from here, then their allele frequencies are related, not only because of the fact that they are in similar kind of a geographical region, but it is also related because this population might have an ancestor with this population, or because of the geographical proximity, there might be migration between them or a mixture between them, which makes them very similar looking to one another. So just looking at linear trains, so there might exist some kind of linear trains in our genomes in populations which are not related to anything which is environmental, but it's just because this relationship or this correlation in this allele frequencies exist because some of these populations shared common ancestry, some of these populations are closely related, and are also oftentimes have migration and admixture between them. So a linear relationship is so we can look at really how y is affected by x, but we really try to build a null model of what kind of expectations we have in allele frequencies in two neighboring populations, or what kind of differences do you expect, and if there is a difference with respect to allele frequencies, if one of these population is going through some kind of a selective pressure, which the other one isn't. All right. So to put this concept into this, there has been various kinds of methods which were there in the literature. The most prominent and the most widely used of this was something which is called a PC-Adapt, which is an R package to perform genome scans of selection based on principal component analysis. This was published in Molecular Ecology Resources in 2017. And then there were another resource which was which also there. Now, what we did is we moved from a linear setup to a more complicated setup, or a more intense setup, which is a deep learning setup. But the reason, of course, people would ask is, why did we do that? Now, one is which I just mentioned in the previous thing, is that there exists some kind of a correlation structure in the allele frequencies of neighboring populations, and there is some kind of a null distribution from which you want to separate them. And the psi x, or the relationship between y and x, is very, very complex. And the factor affecting this are unknown, and it's very, very large. So it is not necessarily we thought that it may not be just something which is like a linear or a simple quadratic relationship between y and x, which might hold true. So there might be a requirement for identification of complicated patterns which exist in the data to try and infer about y. There was other advantage, which is a general advantage in AI, ML, and deep learning kind of methods, which is that the genome, although the genome being large makes it more difficult to find the signals, the genome being large also makes it more important and more beneficial for us to use these large data sets in the data intensive AI, ML, or the deep learning methods. And the advantage of using a deep learning method is that the genomic data is continuously being generated. So there is almost like a reinforcement, which even if you want or do not want, is continuously going to happen. So as we go and generate more and more data, we are going to improve our models, not only the fit or not only make it more robust, but our models are only going to improve over and over again if we can build a flexibility of that through deep learning in our model. So having said that, we are not the first or the only people who have thought about this. There has been methods where people have tried to use deep learning in trying to answer the same kinds of questions, and there exists a method which was published in these papers. This is called a deep genome scan, and we'll get into a head-on comparison between our methods and the linear models, and as well as deep genome scan. All right, so here is our model, a brief description of our model. I'll obviously, as I mentioned, I'll not get into the gory details of the mathematical framework of this. So our model has two stages. The first part is a hyperparameter optimization. So what we do is we have a genotype count matrix and an environmental factor matrix. These are very, very flexible. We can increase the sample sizes, you can increase the number of populations in which you'd like to make these comparisons. The environmental factors depends on whatever kind of information that you can gather on this environmental factor matrices, and all of this can be put into an optimization algorithm by which we find a best artificial neural network architecture. You do not have to do too much, but if you give it a genotype data, which is very, very large in content. For instance, if you can give it, suppose, something like a whole genome sequence data, then this program will automatically take random chunks from this genome to optimize this, such that we quickly get to the best ANN architecture, or some kind of an optimized ANN architecture by optimizing on this hyperparameter space. So one of the major breakthroughs which our method could achieve is because we did a lot of work and a lot of research on this hyperparameter space, and we have actually developed a algorithm, a combination of previous existing methods into the optimization process of this hyperparameter space. Now, once you have the best ANN architecture, you can use your, so this is like something which is like a training data set. You can train your ANN architecture with the entire genotype data, not from the random regions now, but some segment of or a large chunk of individuals on the genotype data. You can train this, you can weight the matrices of the ANN, you can do outlier features detection. This is another thing which I'll go into the next part of my talk in a little bit, and then eventually you can find the outlier genomic regions, which are the regions under natural selection. So another important theoretical change which we have done in this particular architecture of ours is that what has generally been a criticism of this deep learning methods is that they act like a black box. So you put something in and you get something out. You do not know what it is doing as a black box, and you can see that the output is fitting well with the data, but you do not know what the reason is. Why is it fitting very well, or what might? So in our context, you might find if you give the genomic data set and you get some regions which are selected, you do not know which environmental factors. In a black box, you won't be knowing which environmental factors are actually impacting on this output. Now, what we have done is we have done some kind of a mathematical, some mathematical formulation of ways and means by which we can have some sense of the features which are being selected. Or in other words, we can estimate the contributions of some of these input factors, which may be environmental, which may be environmental factors which are interacting with the genotypes of individuals. All right. So our research hypothesis, which was to find the selection gradients across this, and the objective, which was to detect signatures of natural selection, now have these research questions where we find the environmental factors, because we have this feature selection where we can also find the environmental factors that drive this natural selection. And then there are spatially, if you have enough data about this, then we find the phenotypic traits which are under selective pressure and the genetic variants, particularly this is the first part, the genetic variants which are responsible for these adaptive traits. Now, when you develop a model, the model might theoretically look good, but you have to compare it with head-on with the other kinds of existing models that are there in the literature and that are there in usage. So we used a model which was developed in this particular paper, and this model was the gold standard by which these kinds of methods were tested against one another. Let me run you through this model. So what we have is like a chessboard, a 8x8 grid. So there are 64 populations. In each of this grid, there will be populations, and the climate or the environmental exposure will change across this x-axis and the y-axis. So there will be gradients in the x-axis and the y-axis. So in each of them, we have populations of size 200 with initial allele frequency. The starting allele frequency is 0.5 for all of them. There is some kind of a recombination and migration rate between this. So these lattices are not like fixed places, but there has been migration with adjacent lattices. And then we do this or develop this simulation as it has been described in this 2018 paper on just 1,000 individuals. All right. So, sorry, 1,000 loci. So the model here defines the fitness of individual based on 3 different quantitative traits: QT1, QT2, QT3. Each of this is polygenic, which depends on 10 independent loci for each of this. So each trait is determined by 10 independent loci, and there are 3 traits: Q1, Q2, Q3. And fitness for the ith trait is something which is, because there's a quantitative variable, the fitness is some kind of a function which is like a normal function. And the overall fitness of an individual is the fitness from the first trait multiplied by the fitness from the second trait multiplied by the fitness from the third trait. So every individual has all of these traits. So this is more like trying to mimic a situation which is typical for a polygenic quantitative trait, where multiple traits give rise to certain kinds of fitnesses. And eventually, what we do is we do 500 generations. So under different kinds of Q1, Q2, Q3. I'll immediately tell you how we have varied the simulation gradients over this axis. And we generate this population for 500 generations, sample 10 individuals, which results in 640 individuals for with 1,000 loci, and try to find this 10 plus 10 plus 10, this 30 loci, which are actually the loci which are causing these changes, right? So this is a simulation, so we know what the loci is. So we know where the loci are, so we know what the correct answer is. So for the first trait interacts with environment where the environment changes in a quadratic fashion. And the more to the red is, which means that there is more impact of the environment on the trait. This is for the first trait, and the second trait varies in a linear fashion. QT3, this varies in a linear fashion, and then there is Q22, which is in a patchy fashion, which is like a gradient of environment. This is typically what you see in life, that there are patches like heat changing from one direction to another direction, humidity changing from one direction to another direction, in a relatively continuous fashion. The most difficult of them is here, where it is almost random. So the QT1, or the first trait, is interacting with environment in a quadratic fashion. QT2 is environment interacting in a linear fashion, and QT3 is interacting in a patchy fashion. So when you try to find the underlying loci for these traits, keep an eye on raising, which is our method, which is in green. And here we are looking at the true positive rates. And as you can see, that green, the raising has a higher true positive rate for Q21. It determines Q22 with almost 100% certainty, and in Q23 it determines it much better than the PGLS or the existing algorithms, existing linear and other kinds of non-linear models of regression that we have, which are not AI driven, right? So we do much better on the true positive rate, but this is typically known for artificial intelligence, machine learning, and deep learning algorithms, that they typically do good for true positive rates, but oftentimes do this by overfitting. So what we look into this is looking at the false positive rates, and we also find that raising also does good in false positive rates, but it does best in the false discovery rate, which is comforting. So when we compare the deep genome scan with our proposed network, again, on the left, the red is our proposed network, the artificial neural network that we have, and this is deep genome scan. In order to do this simulation with deep genome scan, this is the execution time in hours. So our method took 2 hours, while deep genome scan took 121 hours. So obviously, a lot of simulation with deep genome scan was impossible, but as we can see that we perform better than deep genome scan in both identifying the loci for QT1, QT2, or QT3. So either way, deep genome scan is not efficient. It is taking somewhere around 60 times more time than what our method is taking, and it is not performing as good as we were. So this was the existing method. This is the existing simulation in the world with which people compared, but we did something which is a little bit more complicated. So we had a demographic model where we have 25 populations. So this is the first model, which is like a grid, where there are different kinds of exchanges between all of this. But we also developed 2 other models, a demographic model 2 and a demographic model 3, which is more or less like mimicking the human population, in a sense that this model has a root from which there are populations which are derived, and one of them remains in one place, and there has been exchanges between this. This population becomes an ancestral to 2 different populations. This population becomes an ancestral to population 2, as well as gives rise to population 4 and population 5. So in your y-axis is your time. So it is a same population at this time point, which is 20,000 generations back. Then it gets to different kinds of populations. One population remain here. So there are some populations which are more closely related, which happens in a more recent times, where the fissions and the fusions happen more recent times. So this is where the population sizes remain same. And this is another model where we have changed the population sizes, some of which has gone through contraction, most of which has gone through expansion. These are all mimicking what we know about the human population demography that we have. So what we did is, under this demographic model, we generated data. Now this time the data is generated in a genome-wide scale, not only on this toy data set of only 1,000 individuals, but at a genome-wide scale like the human genomes, and we tested all of these existing methods. So I'll not get into the details of all of this. What I'll try to tell you is that our algorithm is in red, raising is in red. So the red lines are raising. The PZ adapt and the ridge are in the blue lines and the black lines. So on the top panel under the different demographic model, the first one is the grid model. The second one is the model with where there are these fissions and fusions happening, but the population sizes are not changing too much. And the third model is the most complex one, where the population sizes are changing hugely. As you can see, this is the true positive rate on the top panel. The red is our method, where the true positive rate uniformly is performing much better than the blue line and the black line. Particularly here, when there is a linear effect of the linear gradient of the environment, which is impacting on the selection, and our method distances itself from everybody else. If you look at the false discovery rate, you want a low false discovery rate. So you want the red line to be at the bottom now. As you can see, the red line is at the bottom, particularly when the demographic model becomes more and more complex. So our method really performs well as the models become more and more complex, and it performs way better than everything else. We could not assess the effect of deep genome scan, because deep genome scan is so time consuming that it really cannot be, and there is no way in which deep genome scan actually can be applied on something which is in a genome-wide scale. So what we then did is that after the simulations, and we're convinced about the simulation that our method is performing well, we went to real data and applied this on the genome-wide population, genome-wide implementation. So what we did is that we did some kind of hyperparameter tuning by choosing this regions from different populations, and did this hyperparameter tuning to get to this ANN architecture. And once we had this ANN architecture, we devised another sliding window approach by which we looked into small regions around a genomic region and slided our window to get something which is like a composite over that region, such that we can generate this in bits and pieces and then make it continuous over this. So what this gave us, this implementation gave us, is an advantage by which we can parallelly apply ANN into different so this is a program which is very parallel friendly. So you can parallelly apply to this into multiple machines and then combine all of them to come up with so it has an inbuilt parallel architecture inbuilt in them, by which you can do it, apply it parallelly, divide and conquer, then put everything together to come up with a combined estimate. So when we applied this on the peopling of the globe, this is what we found. And there is a prelude to this, as we have said, that we have lived in Africa for a very long period of time. More than half of the time we're on this earth, we have spent in Africa. And then we moved out from Africa in a small group of individuals, lived here, and then moved to different directions, one to Southeast Asia, including India, another to East Asia, and another to Europe. These are the ancient migration routes depicted in the dark green. And that is the reason that we looked into the genomic regions, which are in this continental groups, or regions which are in Africa, Europe, and Southeast Asia and East Asia, South Asia and East Asia, and try to find which regions of the genome has responded to environment or has been selected by environmental pressure. As you can see, the size of the genomic regions in Africa are smaller. The size of the genomic region everywhere else is more or less. The distributions are more or less similar, gives us confidence, because Africa is a place where there has been a lot of recombination. These are more ancient population. There has been a lot of recombination. So things have arose earlier, and because of recombination, they have been broken into small chunks. So the average genomic region in Africa is smaller, whereas because they have history of similar kind out of Africa, the history is more or less of similar antiquity, similar time frames. So their distributions are more or less similar, which is very reassuring to know. And also, if you look at the shared regions for this, shared regions among this. Then there is another thing which comes out, which is striking, which is that the African populations have a lot of these selection signatures which are unique to Africa, whereas the selection signatures which are unique to the Europeans or the South or the East Asians or the Southeast Asians are much smaller in proportion. So all of this also depicts in understanding our history, and it is what is expected under the history. We have done some kind of a dissection of what this might be, but this is not I do not have the scope, and this is also an ongoing work, so I do not have the scope to elaborate on what these regions are and what these selective forces are. But this is what we got. So in summary, the implemented artificial network demonstrates both robustness, time efficiency related to existing methods. It is much better in head-on comparison in simulated data sets, way better in terms of time, way better in terms of finding the true positives as well as keeping the false positives in check. Among continental groups, Africans exhibit the highest percentage of unique natural selection signatures, and non-African populations show a higher degree of shared genomic regions. So with that, I'd like to end this talk with acknowledgement to all the people in the world who have, over time, I'm a population geneticist, I always feel very indebted to these people who have contributed to our studies through their blood samples, DNA samples, and their consent to participate into this without looking into an immediate profit. And to the Department of Biotechnology who supports our institution and myself. Thank you.
Thank you, Professor Basu. For our audience, online audience, you can always type in your questions in the chat box on YouTube, and we'll be able to address them now. Maybe we can wait for a few minutes if they have any questions. I think I just have a naive question for you. In the penultimate slide, where you showed the pie charts for different populations, and of course, you've mentioned that there are larger common unique regions in the African populations. Why? What is the reason behind this? Why is it that that population has more unique regions and
yeah, I mean, I think so. I mean, obviously, I think and we think that this is because of the long unique history of Africans. So in a sense that if you think about the human evolution, so if it has all started in Africa in about 200,000 years, people have lived in Africa for the first 150,000 years, right? So this has a very unique history which Africans have. And there must have been changes in the environment to which Africans did respond. Now, this is something which is not there for anybody else. So these African individuals have this, which is grind in the genome, and some of these will still be retained. And there are these specific regions in this which has risen in very high frequency in Africa. But when you start moving out of Africa to different directions, these genomic regions are not forced under the same kind of selective pressures anymore. So those can be either utilized for certain something else, or the genomic signatures eventually gets erased from those regions. So on other populations, these particular regions do not need to be selected anymore. So with the course of history, the signatures typically would get eroded, and that is possibly what we are seeing here. Did that answer your question?
Yes. Thank you. We have a question from Dr. Priyadashi Basu. Um, as examples of both spatial and temporal, you gave the example of pathogen exposure. How are they different? You are muted.
Yeah, I mean, what I said is that there are selection gradients, some of which are spatial, which is geography and altitude, and then there are pathogen exposure, which if you just think of this in a spatial terms, then the pathogenic exposure is spatial in a sense that the pathogens also optimize them with respect to geography and altitude, with respect to the geographical or with respect to which place we are in. Similarly, but these pathogens also change with time, right? The pathogens change even in a region. The pathogens do change with time. So what I'm trying to emphasize is it is not necessarily different. What I'm trying to say is the selection gradient is actually both spatio-temporal. It is spatial. We have a sense that it is spatial, and it is also temporal because it can oftentimes change with time. And these are not, again, unrelated to one another because spatial changes do give rise to temporal changes as well. Like the example of agriculture, where the spatial changes in specific region give rise to different kinds of changes which has happened over time, et cetera, which leads to changes in human intervention, changes because of human intervention, environmental changes because of human intervention, and as well changes in the pathogenic spectrum, et cetera, et cetera. So yeah, it's not different, it is intertwined, and that's the reason it is more complex. So that's the reason selection gradient is more complex, and that is also the reason why if you try to write something like a y equal to psi x, your x's are very, very strange, and the relationship of x and psi is also typically very, very difficult. And that's one of the reasons, that's one of the intuitions why we think that the artificial intelligence, machine learning, or the deep learning methods are much more suited for an application of this kind.
Okay. We have another question from Kallav Datta, and he says, excellent work and excellent talk. Can you please explain one more time how you are partitioning the genomic regions and how the moving window approach is helping?
Okay, so let me try and do this. So what we did was that, so see, I mean, the first part, of course, is to define the is to define the architecture. So that we did with different kinds of subsets from different populations from which we find this architecture. Now, once we get this architecture, you can apply this architecture on different segments, right? So this gives you the advantage of applying this in parallel, right? So you fire this in parallel. So you get this in different segments all across this. Now, the problem is that there might be regions which are in the junctions which are not adequately represented if you have done this in windows which are disjoint to one another. So what we try to do is we try to smooth it off. So this is essentially something which is like a smoothing experiment where you have the segments, and on the segments you have this this window, which you move around along this genome, and such that in some of these, so in order to get the idea from this region, you get windows which are here, and you get windows which are here, and then you use something which is like a smoothing function for this to come up with something which is true for the entire genome. I hope this this answers your question. I mean, we can get into the technicalities of this, Kallal, if you when in a discussion which is like more technical discussion. I do not want to get into that kind of a technicality in this talk, because that will be pretty equation intensive and eventually possibly not very interesting to most of the audience.
So, and one last question in the chat box is from Shavanik Singh Gupta. He asks, how can we incorporate the existing biological information and phenotype information available in the models?
Yeah, so what we do is we go it in a pretty agnostic way, in a sense that all of this gets into as an input, and it is all treated with, when it gets into an input, nothing is given importance or a relative ranking of importance. So anything which is even unrelated can get into this. So there is no harm in giving something which is even unrelated to this. What will happen is that the program will eventually decide on which environmental factors are actually important, like it will decide which genomic factors are important here. So this is the advantage of using this again. This is the power of the deep learning method here, that any environmental factor that you think important for which might impact selection, which might impact the genomic architecture or the pattern, can be incorporated here if you have the data. So if you have the data for that population, anything can be incorporated here. The issue is the advantage is that the program will take into account this and try to use only the ones which are important to it. Now the problem is that if you put too much of an input, then obviously it will take a much longer time to run, because it has to train itself to find out the ones which are not so important.
So, well, thank you, Professor Basu. I think we have answered all the questions which were raised in the chat box. And if you have any further questions, Professor Basu has already shared his email ID, which is ab1@nibmg.ac.in. So please do feel free to drop in an email for any further queries. We thank you for joining online. And also, just a heads up that we will be organizing more webinars in this series, and which showcase all the work that is being done at NIBMG and at other IBRIC Plus institutes as well. And thank you, Professor Basu, and congratulations again on this fantastic work.
Thank you so much. I'd also like to take this opportunity to thank IBRIC and DBT for organizing this process to showcase our work. I'd like to thank you for organizing this, and Professor Sagar Sengupta, our director, who has also been very, very encouraging to and essentially did not give me an option but to give this webinar. But finally, big thanks to all people who are listening to this webinar and asking questions and listening to this. As we said, our program is available. Please do use it. If you have a problem or a similar problem, do use it. Or if you have questions, write to me or the appreciation. Thank you so much.
Thank you. Thank you, everyone.